Hi everyone. In this experiment, we're going to determine water hardness in Duck Pond and Boone Creek by titration with EDTA. EDTA stands for ethylene, diamine, tetraacetic acid. And the method that we're using is actually a standard method. It's actually method 2340C. And a lot of our experiments this semester are going to be from this book of standard methods or EPA methods. So if you go on a job interview and they ask you, do you have any experience either with standard methods or EPA methods, you could say that you have some. So a little bit about EDTA, it's found in all sorts of these health and beauty type products and even some food products, but today it's going to be a titrant in our experiment. So our EDTA solution is going to be in this burette, and either our Duck Pond or Boone Creek sample is going to be in the Erlenmeyer, and Hardness is going to be our analyte. So why do we even care about water hardness, and what is it? Well, hard water has elevated calcium and magnesium concentrations. And why is that bad? Well, basically it binds to soap and prevents it from doing its job. Here's a chemical equation demonstrating that. This substance here is a generic soap molecule. And soap is a surfactant, meaning that it has a so-called polar head group and a non-polar tail. So this alkane could be anywhere from, say, 10 to 18 carbons long, making it non-polar. And why is this substance such a good soap? Well, the polar head group makes it soluble in water, and the non-polar tail does a really good job of carrying away oil and grease and other non-polar dirt out of the water. But as you can see here, when your water is hard, the calcium or the magnesium binds to the soap and it precipitates out, forming soap scum. So I'm wondering, where does this calcium in water come from? What is the source of it? Well, oftentimes calcium gets into water naturally. It's found in a mineral called limestone and lots of other minerals. So because limestone and other minerals are the main sources of water hardness, if you think about what the formula of limestone is, it's actually calcium carbonate. So the units for water hardness are milligrams per liter, or more clearly, milligrams of calcium carbonate for every liter of sample or solution. This is our titration reaction. So for every 1 mole of calcium, 1 mole of EDTA binds to form a soluble calcium EDTA complex. Now, I'm not going to go over the calculations in gory detail in this video, but I'll just try to get you started. You will know the molarity of your EDTA solution, and you will know the volume of EDTA solution used, remembering that molarity is moles per liter. If you multiply by liter, that'll give you the moles of EDTA used in the titration. And looking at your stoichiometry for every 1 mole of EDTA, that's 1 mole of calcium ion, and thinking about the units for water hardness for every 1 mole of calcium, That's 1 mole of calcium carbonate. So remember, we're trying to get to milligrams of calcium carbonate for every liter of sample. So we can convert moles of calcium carbonate into grams of calcium carbonate, and then milligrams of calcium carbonate. And then remember to divide by the volume in liters of sample that you titrated. Let me show you some common mistakes I've seen. Sometimes students don't use the formula weight of calcium carbonate. They just use the formula weight of calcium when converting from moles into grams. So remember to do that. Sometimes students forget to divide by the volume of sample titrated, and sometimes students incorrectly calculate the concentration of their EDTA solution. So watch out for those 3 errors. Just to make sure that you're on the right track, let me show you some historical data. So these values of Duck Pond water hardness and Boone Creek water hardness from the last 3 or 4 semesters should give you an idea of what to expect. You can see that in the spring semesters, Boone Creek's water hardness is a little bit higher. And notice that we get in most semesters pretty good reproducibility. Anytime we have poor reproducibility, 9 times out of 10, it's caused by a calculation error. So in doing this titration, how do you know when to stop adding the EDTA to your Erlenmeyer? Well, you're looking for a color change caused by an indicator. And the indicator that we're using is EBT, or Eriochrome black tea. So what you'll do is, to your Duck Pond or Boone Creek sample, you'll add a couple of drops of EBT, and your solution is going to turn a wine red, kind of shown here. And then as you start adding the EDTA, it displaces the EBT from the calcium to form our calcium EDTA complex and free EBT, which is a true blue color. So you're looking for a color change from wine red to true blue. As you start getting closer to the endpoint, due to the presence of both bound EBT that's red and free EBT that's blue, your sample is going to turn purple. But this is not the endpoint. You want to go all the way to that true blue. So again, wine red to true blue is our color change. And what are we going to do for quality control in this experiment? So again, is there something that we could titrate with a known calcium ion concentration? That would be a good thing to do for quality control. And that's exactly what we're going to do. So we're going to titrate a solution with a known calcium ion concentration. You prepare it, and I think the concentration is 2.5 millimolar. And this solution is called an LFB. LFB stands for laboratory fortified blank. So in other words, it's a blank or deionized water that you fortified with a known calcium ion concentration. So when it's all said and done, you're hoping that you can tell the customer that you got a value close to the known concentration. So some reminders: mix all solutions well. I cannot stress how important this is. Even if you've prepared a solution, you've mixed it, and it's sat aside for only 5 minutes, mix it again. You won't believe what a difference this makes. The procedure tells you to dry the EDTA in your desiccator. So you'll weigh a little bit out in a weighing bottle, and then you'll put it in the desiccator to dry for an hour. But should this cap be off or on in the weighing bottle? Well, the point of drying the EDTA is to remove excess water, and if the cap is on, that isn't going to happen. So the cap should be off when your EDTA is in the desiccator. And just something to remember about the desiccator, try to keep the lid on as much as possible. If you take this lid off and leave it off, all of your desiccant is going to get saturated with water vapor from the atmosphere, and you don't want that to happen. I think the procedure says to dissolve something like 0.6 grams of dried EDTA in a 500 ml vol flask, and it says to quantitatively transfer the EDTA to the vol flask. Now, what does this mean? This means that you want to get every little speck of EDTA from your weighing bottle or your weigh paper into this vol flask. So I've seen some students use a funnel just to make sure that they don't spill any, and then they wash all that salt into the vol flask using a wash bottle. And then this EDTA salt, once it's all in here, won't dissolve in room temp water. You've got to use hot water. Not boiling water, just hot water, so maybe pour some hot water, maybe to there. So swirl the flask to get your EDTA to dissolve. Allow it to cool to room temp, and then dilute to the mark with deionized water. And what mark do I mean? Well, you can't see it in this picture, but there's a mark here that you can dilute to using deionized water. So the main thing I wanted to emphasize here is this idea of a quantitative transfer, and just the fact that the water needs to be hot, not boiling. You'll work individually in this experiment. You will do either Duck Pond or Boone Creek. I'll make those assignments in class. Try to titrate your laboratory fortified blank 3 times and the sample that has been assigned to you 3 times. If you can only do one of these 3 times, I would give priority to the LFB since that's our quality control. The main safety warning in this experiment is the pH 10 buffer, which has a strong ammonia smell. So take only what you need and try to keep it under your hood as much as possible. There are some solutions that you actually can wash down the drain. That's your excess calcium stock or any excess LFB. All other liquids should go into the liquid waste container, and the only excess solid that you should have is the EDTA, and we'll have a solid waste container for that. And for next week, be ready to share your Duck Pond and Boone Creek water hardness values with the class, and then I'll ask you to see the syllabus for the other reading assignments that you have to complete. No report next week, just we want to talk about, I want you to do your water hardness calculations on Duck Pond and Boone Creek for next week. And just as a reminder, this experiment, the calculations do not involve a calibration curve. So that's all. Thanks for listening, and I'll see you in lab.